The overall goal of this assay is to detect autoantibodies against the NMDA receptor in the blood of patients with suspected autoimmune encephalitis.
The method helps answer the key questions in the differential diagnosis of patients with acute neuropsychiatric symptoms. The main advantage of this technique is that it is a simple and reliable screening method.
Begin this procedure by preparing gelatin-coated culture plates. Aliquot 200 microliters of a 2% gelatin solution to each well of a 48-well culture plate, and incubate the plate at 37 degrees Celsius for at least 30 minutes. After 30 minutes, aspirate the gelatin solution from the wells. Seed in each well 5 × 10 to the 4th HEC-293 cells in 200 microliters of cell culture medium containing 10% FBS. Incubate the plate overnight at 37 degrees Celsius in a humidified incubator supplied with 5% carbon dioxide. On the following day, replace the spent culture medium in each well with 200 microliters of fresh culture medium containing 10% FBS. Scaling up according to the total number of wells in the experiment, prepare solution A by mixing the NR1-GFP plasmid with culture medium. Prepare solution B by mixing the transfection reagent with culture medium. Prepare solution C by mixing solution A and solution B and incubating the mixture at room temperature for 20 to 30 minutes.